you all there. Well, it's 9:30. Welcome to Neuroccino. Lovely to see you all here. We have at least 3 papers today. So for those of you who are presenting, if you want to share your links in the chat, that'd be great. Rule of the games. You all know them, but let me quickly repeat them. Briefly present the paper, tell us why you got excited about it, and then we can have a little chat about the paper. That's it. Without further ado, I hand over to Hervé for his first Neuroccino paper.
Hello, everyone. So I'm putting the link in somewhere in the chat. Yes, just now. Okay, so hello, everyone. So I will present a paper. So let me share my screen. Can you see it?
Yes, lovely. We shared some new features on Zoom.
Okay. Okay, let's go back to the beginning.
That's going to be interesting.
Yes. Okay. So the paper is about fine-grained temporal mapping of derived high-frequency variants supports the mosaic nature of evolution of Homo sapiens. So it's a paper about coming from the group of Cedric. I don't know how to say his name. Box, I think. So it's Alejandro Andirco. First of all. It was a trap. So last Sunday, Michel presented me this paper. I say, oh, great. Oh, nice paper. But I didn't see Stephanie was just behind and say, oh, yes, great. Present it to the Neuroccino next Monday. I say, okay, I'm trapped. So let's do it. So, in fact, this paper is particularly interesting because it's merging some, it's about genetics. So you will see it's much more about genetic than neuroscience in general, but it's about evolution, and I'm pretty interested about genetic, evolution, and brain. So it's why it seems this paper has some focus on that. It's why I'm presenting it today. So some disclosure, let's like that. I'm not a geneticist, so I'm interested in imaging and in some approach called neuroimaging genetics. So where you merge neuroimaging stuff and genetic. So I will try to describe this paper. So maybe I can make some mistakes, but okay. So I think I got the main goals of this paper. Okay, so why this paper is interesting about within the topic of neuroscience? I think when you are interested about brain evolution of Homo sapiens, you have different ways to approach it. The first one is the direct way. So you compare the brain, the actual brain of the current population, and you compare it to archaic brains of our ancestors. And the only problem with that, so we cannot do it because we don't have fossilized brains from this period. So we can only derive some, for example, in this picture, you can derive some shapes of the brain based on the endocast. So it's great, but not enough. The second way is to compare with our closest relatives, the primates. So you can take the brain of the primates and you compare it to our brains and you see if they are different or not. And the third way is to look at the genetic. So you do kind of a paleogenetic brain. So you extract information in our DNA, that belongs to our ancestors. We have in our genome some part of our ancestors. So we can use that. And as you know, the brain is coded partially, not completely, but coded in part by the genetic code. So this is an interesting approach. And this paper is about that. So what they did, they took all the genetic variants distinguishing modern humans from archaic hominins. So to do that, the first way the way they didn't do it, you take the fixed differences. So you take all the variants, and for all these variants, you take all the variants for which the current population, so the present day humans, carry a derived allele, so the new allele, while at least 2 of the 3 archaics carry the ancestral alleles. So that means it's a very binary approach. So all humans have the new allele, and the past, our past species have the ancestral allele. So very binary, and I will go back to that later. The other approach is to take the high frequency differences. So it's a more global, it's taking more variants, because it's take all variants with 90% of present day humans carrying a derived allele, and while at least the Denisovan or Neanderthal carry the ancestral allele. The reason they did that is, first, you have more variants than the fixed differences. The fixed differences is very binary, as I said, and there is less variants like that. So with the high frequency, you have more variants to look at. And the second reason is they used a tool. This is the GEVA tool, meaning genealogical estimation of variant age. And for these tools to work, it needs a high frequency. At least it needs variability in the variants. So it's why they took all the high frequency variants. Okay, so they use this tool. I will not go in detail because I don't know the details of this approach. The only what I can say, it's derived it computes and estimates the age of the variants. So based on the current frequency in the population, and based on what we know about mutation. So for example, we know that in average, all humans have 70 variants compared to the parents. So we know the current speed of the mutation. So based on that, you can derive kind of, so you have the speed and the current frequency. So you can derive age of the birth of these variants. Let's say that like that. Okay. So you have the your variants, which make different from the which make us different from the archaic brains and archaic species, and you have the age. So very simple approach. You do an histogram. So here you can see you have the variants here on the variance on this side, and the age on this side. Okay, and you have the distribution of the variance of the high frequency variants. So what you can see, it's a very bimodal distribution with a large peak at the beginning, so and a second peak around 100k years. Okay, so from that they decided to do 3 groups based on 3 specific periods, based on the literature and milestone of our history. So the first one is about emergence of Homo sapiens, going from 0 to 300 key years. And the second one is being earlier mem of our species. And the third is a more recent common ancestor with Neanderthal and Denisovan lineage. So you have these 3 groups of variants based on the age and on these 3 periods, and they went a little bit deeper in the trying to explain what these variants were doing. So the first thing they did, they took all these variants. So these variants belongs to genes, and genes make functions. So the gene ontology is a database where you can link genes to a function. So they took these 3 groups of variants, and they group all the function based on the age of the variant. So you can see these 3 groups, and they derive some function of the genes in these three periods. So you can see some stuff about neuroscience somewhere. Let's say where the postsynaptic membrane potential, presynaptic active zone, and some stuff like that. So nothing very specific. This is a problem with this kind of database. It's not very specific, very large function. So they continue. So what they did, they look at this time they took all the variants and they look at the predicted gene expression. So the effect of these variants on the expression of the genes based on the messenger RNA in the brain tissues. So here you have not anymore 3 time windows, but 6 time windows. Okay, here on here you can see my yes, okay. So here you see the time. Here again you see the expression of the genes related to the variants. So here it's only about brain tissues. So we are closer to neuroscience in this picture. So the first thing they discuss is about this gap between 200 and 300 kilo years. So what they discuss about it is they say it seems to be that these periods correspond to a significant behavioral shift in our lineage based on this gene expression. So it's a big change between here and here. So another thing they stressed out in this figure is the brain cerebellum and the brain cerebellar hemisphere. So the expression change in the cerebellum. I will come back to that later. Finally, to be closer to the brain, they went back to the UK Biobank brain phenotypes. So they took the variants they got and they look at the association between these variants and the brain phenotypes from UK Biobank. And what they found some hits and some association. They were pretty happy about here again the volume of gray matter in cerebellum. And also the precuneus, they talk about precuneus, but I didn't see it in the paper, maybe in the supplementary material, but I didn't see it. And the reason why they were pretty excited about the cerebellum and the precuneus is because they know there is this phenomenon of globularization through the evolution of the brain. So about the shape of the brain compared to our ancestors, there is a change of the shape of the endocast, and mainly in the cerebellum part and in the parietal part. Also a little bit on the frontal, but less. So I think I'm done. So the conclusion was that they made the link between these eye variants and something about the brain shapes and brain function, brain structure at least. And thanks for your attention.
Thank you. You clearly had a lot of time to prepare this. Proper slides.
No. Inspiration, only inspiration.
Well, thank you very much. Are there any questions? No one. So what's the big wow? Why were you so excited about this?
I think excited. I don't know. Let's see. So the reason why I was excited by this this paper is because I'm trying to use this kind of approach to tackle some brain phenotypes. So you can really take you can select some part of the genomes, they are clearly identified as related to our ancestors. So we don't know really the function of these variants, but they evolve to have these variants. So you can really link these variants, this variation of genetic with brain phenotypes. So it's still in the approach of neuroimaging genetics, but this is really exciting to have this possibility to go back in time. It's like the in a movie, you watch a movie, you have the current scene, and you say, oh, yeah, I want to go back. With the brain, it's not clearly possible, but with genetics, it seems to be more possible to go back like that in different period of time. So that was I was really excited, and Michel knew it. It's why he trapped me and he got me in this paper. But that was yeah, I was happy to do that. So thank you, Michel.
I'm very glad you made it and you presented it to us.
Yeah.
Leah has a question.
Yes, I wanted to ask, thank you for the presentation. I wanted to ask if like variations is something that you usually look in this analysis, or is a new approach before look to correlations?
No, in fact,
I don't
it's very old story. Variation is so a SNP, a variant, is you have two alleles. It's kind of simple. It's complicated at the same time. When you look at the variants, it's very binary. So you are homozygotes, heterozygotes, or non-carriers. So you have three possibilities. So you look at that. So it's a correlation with 3 points, and you can do that with brain phenotypes. So it's become more complicated when you have more variants and when they are linked together. So you do haplotypes to get the full picture of the variation, genetic variation. So, but basically, it's very simple at the start. So it's why after you can really look at very simple correlation and go deeper and more complicated in a way.
Okay, thank you.
Michel, you're unmuted. Did you want to say something?
Well, yeah, it's like mostly I was saying we really should do like a whiteboard or a focus group on your
Yes.
project. That'll be cool to
Yeah.
think like how to tackle this
Yeah.
big question, which is a link between genes and the brain. And
yeah,
that'll be great. Let us know when you're available.
Yeah, I will tell you because 2021 will be yeah. I want to start by that. So
and then now we say, you know, to the evaluation committee that we will do it. So we have to do it.
We have to do it in a way or in another. So let's do it.
All right, cool.
It's going to be great. Right, any more questions? No? Okay, let's move on to the next paper. Maybe we can even fit in all 4 we have today. Leah, do you want to go next?
Yes.
Take it away.
So I'm gonna share. Okay, can you see? I think yes. And so this is a paper published last week in Brain. And I thought it was really interesting because is trying to link neurodegenerative diseases with psychosis symptoms. This paper is by Georges Nizam, I think, et al. And colleague, and is a retrospective study. They select patients that had pathology, and so they were really able to classify with kind of neurodegeneratives where was going on. And so they select 372 patients, and they screen all the patient's medical records to see if there was some manifestations of any kind of psychosis. And actually, this psychosis was present in 111 patients, so a high rate of those patients. In particular, they divided in different groups. There were patients with Alzheimer's disease. Maybe I can go to the table. So there were patients with Alzheimer's disease, patients with Lewy body, and also mixed Alzheimer's disease, frontotemporal lobe dementia, including where there was like tau presence or TDP, TAR, DNA binding protein presence, and the different subtypes. So in the tau FTLD, you have also the peak disease, the pulse, the progressive supranuclear palsy and cortical-based degeneration. And they like go through all the clinical informations, the clinical diagnosis, the kind of psychosis. In particular, they focused on two different kind of psychosis. There were hallucinations and delusions. So when you are fixing something that is not seems to reflect the reality, but you are stuck in that view. And they also divided the delusions in delusions that were like bizarre, so something that was not really real in any terms, or something that was non-bizarre, so maybe could be like possible, but reality should give you some like tips that is not something that is real. And also in subgroups of these patients, they look into the genetics, because as the authors nicely said in the introduction and also in the discussion, there are actually, I didn't know about it, a lot of studies that related genetics with, so also linking to the paper presented before, with presence of psychosis. So like in the state of art of literature, seems that there is actually a link about psychosis and the pathological substrates of these neurodegenerative pathologies. So here you have a first chart with the rate of hallucinations and the delusions. So they focused on these two groups, and also they looked in this like time spread of these manifestations. For example, hallucinations, as is known, that is like more present in Parkinsonian syndromes and Lewy body disease. And then they look to also really specific features of these psychoses. So if the hallucinations was about a person or something that is present in when you have like Parkinsonians, is that you saw this shape or something in the corner of the eye, something that is not really clear. Also, you can have this feeling of persecutory or a feeling of a presence. You can see an object, or you can have also a misrepresentation of a place. There was in the article they say that there was this patient that he thought that the house was moved in another place, and he kind of understood that. So he believed this, and it was so confusing for him. Or sometimes some patients forgot where the objects are, and so they think that someone told that, and there is this loop that is like moving forward from reality. And also you can have like delusions that are like grandiose, so you like elevate yourself, or also related to love, so some fixation in that way. And so it was really interesting also in the example that the authors represent here in the paper. Let's see. Like yes, here they say that also like in the body, that like your arm is not really a part of your body, but is like a robotic implant that could be a bizarre delusion, or that your wife is replaced by a demon. They were also actually funny, but yes, it's a serious thing. And here you have all these subtypes that they really divided. So they, in this paper, they really try to find really specific features that can describe these manifestations, and they try to see if there was a different occurrence in the different pathological diseases. And actually they were. As you can see here, there were significant differences. For example, in the hallucinations that were more common when you have Lewy body disease and mixed Alzheimer disease, and also they also conduct a linear regression to see if based on the clinical classifications, if they were able to predict the presence of one of these manifestations. And actually, if you have like an high grade, I think 3-5 BRCA for the Parkinsonians, is more, you can predict if you have hallucinations. And also here, in the case of delusions, they were more present if you have the frontal lobe dementia, and in particular the TDP manifestations. Indeed, they also tried to go more in deep, and for example, compare, there was another table here, for example, compare if there were any different in the frontotemporal dementia if you have like the tau presence or TDP. Actually, there was, in terms of the delusions of psychosis, not really in the peculiarity of these delusions, but for example, in the non-bizarre one, they were more frequent in the TDP variant. And so, like about genetics, they just discussed the frequency. There was like a subgroup that was there was another table, like so they were like less people, but is really is with all these variants, genetic variants, and is really well discussed in the final part of the article, because there are a lot of literature going on in this field, and I found it really interesting because if there is really a link between these manifestations and the neuropathology, maybe you can predict before death what kind of pathological substrates is driven the pathologies, and they say that this is really important also for treatment portions, because now you can focalize so certain treatment, and it is important that you can put the patients in the right category to treat as well as possible. And it's also important that we have knowledge of this, because then in the clinical you can include some tests that look in this kind of psychosis. So maybe clinicians should ask more about this. So if you see a shadow, or if you have any kind of delusions, is important. And also, they say that in this study there was not a systematic way to investigate these psychosis features. There was just some notes in the clinical report of patients. So I think that this perspective can really help in the classification and also treatment of these patients, because it's really something that affects your quality of life. Yeah. So this was the paper. If you have any questions, let me know.
Okay, thank you, Leah. First question. Yes, Michelle.
Yeah.
So I was wondering how rich were the hallucinations that were reported, because it's such a wide range of quality of the hallucinations. So do you have any examples? Or I can hear you.
Sorry. So if they were really different between one patient to another, you are asking that? So there was like some that are really specific, like maybe there was one that only one patient had about animals. So sometimes you can have like, yes, they are really like specific for patients, and they try to create these major categories, like delusions, bizarre or non-bizarre, or hallucination of places, person, shadow, or as if they were like on the corner of the eye field. So they try to summarize, but yes, there was a great variety. That's true.
Because so if you like hallucinations, really like the work of Dominic ffytche is absolutely amazing, and he really has this, you know, classification of the different kind of hallucination that you can have that are really beyond just the category, like, you know, places or faces or animals or things, but like people have like reduplication, magnification. He had like all these gorgeous description of the different kind of hallucinations that should be related to different places in the brain, that should be related to different kind of pathologies, and that, I think it's that's why I asked the question. Thank you, Steffi, for providing the name. It's Dominic with a C, though, not a K.
It's more the last name that's tricky to get right.
Yeah. No capital to ffytche, otherwise he gets really upset. Thank you, Leah. That was very interesting.
For sure, there really is. What I really like, yes, because I had this experience that there was these patients when we were doing like the clinical studies that he saw this shadow. It was the Parkinsonian disease, and he believed that it was like the death. So imagine you. So every day the death in front of you, it's so affecting the quality of life, and I think that it's really important to take care about this aspect.
It's called your mirror. That's what it's called.
Okay. Yeah, that's
rough start this morning, eh?
I had a rough morning.
Okay.
Thank you for the question. Leah, you presented a paper about delusions a while ago, if I remember correctly. Do you remember what the prevalence of delusions is? Because this seems, I forgot the numbers now, but it was nearly 400 patients and 100 something had delusional symptoms. That's a huge number.
Yes, the paper that I presented was about delirium, using that spectrum,
Yeah.
but yes, it's also and also that was really the incidence was really high. It was in neurodegenerative disease, and here, so delusions. Maybe I can share again. So here in the delusions, you have like the 16% in AD, 25.9 in Lewy body AD, and then a little bit like less in the frontotemporal lobe in the tau variant, and higher in the TDP variant. So it's like less than the alpha of the patients, but it's a percentage that can represent the sample.
Yeah, I think in the general population, it's like
in the general
percentage, it's like less than 1% or something. So this is.
So you say that it's something that is also present in normal people.
Well, there's a certain prevalence amongst the general population.
Okay.
But that number is way smaller than what we see in your paper. So I was just curious about that. The second question I had
but about delusions is something that, for example, the non-bizarre one is something that also like not so far away from something that also other kind of not the neurodegenerative diseases, but other manifestation. That is true. I also thought about that during the reading.
Sorry, bad timing. The second question I had was about figure 2. If you quickly want to reshare it.
Yes. So figure 2, this one.
That one. Yeah. So later on you said that we can actually use it to like predict subtypes. But when you look at this figure, at least Lewy body dementia seems to be quite high on most of them. Like, it doesn't look specific.
What?
For Lewy body dementia, it doesn't look specific that you could actually use paranoid perception to actually predict the phenotype.
Yes, when they do the regression, actually, the significant findings were about the Lewy body disease, and there was like the all of these hallucinations could be like predicted by that for example, here you see the Parkinson's disease BRCA stage 5 and 6. Can predict the hallucination of animal in patients with Lewy body pathologies, and like all the findings were like as these. So they were really specific to the Parkinsonian. And yes, actually, yeah, it was not so specific, but was related to hallucinations.
Thank you.
I answered.
Yeah. Any other questions? No, nothing on YouTube for the moment. Okay, great. Let's move on to the next paper. I'm quite excited about this one. It's another brain paper, but a historical take rather than a research paper. And Valentina is going to present it to us. It's her first Neuroccino paper for us. Take it away, Valentina.
Hi, can you hear me? All right. So I'm sharing my screen. Okay, so this is a very interesting paper on Thomas Willis. It's an essay in occasion of his 400th anniversary of the birth. And we all know Thomas Willis for the circle of Willis, which is an entire circle at the base of the brain. But this essay is so interesting because it reveals that actually Willis established the basis for modern research, modern science, and medicine. I'm going to tell you why in a bit. So Willis was instructed with classical studies, basically mainly religious and ancient history studies. He had a bachelor and master degrees in classics, so humanities, but he wanted to study to graduate in medicine. But then English Civil War happened in the middle of the 17th centuries, and he couldn't. He started his bachelor degree in medicine, but he couldn't finish it because of the Civil War. And his studies has been cut short. and so he graduated with his bachelor and master degree very later on. But in the meantime, he was licensed to practice medicine. And according to his notes and the notes from his students, he used to go from village to village, visiting patients. It was very caring. And he had this new approach. He was experience-driven approach without any dogma, because in the 17th century, medicine students were instructed with Aristotle or very ancient medicine dogma. They couldn't criticize what they were studying or reading. So he was free to explore the new approaches in very practical approaches in medicine. So yeah, he received finally his doctor in medicine degree. He was lucky enough to live in a very a renaissance of medicine, because in Oxford he was lucky enough to work with Locke, so philosophers, chemistries, architects. It was a very fruitful, started to make fruitful collaboration with other fields. And this is the modern times of medicine, like showing up because he started to have like different hints and collaborations, and they made a group, actually. Yeah, they were called the Oxford Philosophical Club. So he had collaborated with Wilkins, Boyle, Locke, and they actually discovered, for instance, like the fixative liquor for prolong the life of the brain tissue after dissection, or coined the term cell, or made some extremely beautiful illustration of the cerebral anatomy with with the architect. And yeah, Willis is not only the discoverer of the circle of Willis, but also he named several structures of the brain, such as anterior commissure, corpus triatum, optic thalamus, etc. And he studied the thalamus, the anatomy of thalamus, by the observation of post-mortem dissections. He also was the first illustrator of the cranial nerves. I was surprised by that, but and he combined 4 cranial nerves in just 2, so he wasn't credited for the discovery. Yeah, so the novelty of his approach was the observation of symptoms and mapping the symptoms onto the anatomy that he was exploring post-mortem. And yeah, it was, he started a new approach that is valid also today, and that is being studied today. That's it.
Thank you very much, Valentina. It's a great paper. I greatly enjoyed reading it. And I thought it was quite vivid. Like, you could really see how Oxford shaped him and how he shaped Oxford and how it must have happened at the time. Great paper. Are there questions? No? I saw a lot of you get excited in between. It's like, oh. One thing I never knew is that he actually got his medical degree after just 6 months of doing medicine.
Yeah, they can show us his studies. But well, because he chose the right side of the English Civil War. He was a royalty, not a parliament, so it was rewarded for that.
Yeah. And then the PhD was 14 years later.
Yeah, but still, he was practicing in the meantime.
Correct. There were a couple of videos in the paper that I couldn't easily find, but I only skimmed it yesterday. But it could be interesting to have a look at the videos as well. Right, if there is no other questions, thanks for presenting it. We make the link available in the video later on. Michelle, we actually would have time to squeeze you in if you're fast.
Wow.
Didn't expect that.
No, I didn't. Of course I did.
Yeah, 15 minutes.
Let me introduce you to the supplementary I-field tracks cognitive efforts.
Sorry, Michelle, can you zoom in so we can see the paper better?
Yes. Is it good?
Very good, thank you.
All right, this is a paper in rhesus monkey where they used a task of gaze orientation, saccade and anti-saccade, and they did measure the pupil dilatation, which we know is like very reactive to the cognitive demand in tasks. And they also measured the ultrasound imaging in the supplementary eye field and other regions in the brain that I'll show you in a minute. And essentially, so, you know, they've been able to show for the first time the reaction of the supplementary eye field to saccade and anti-saccade. The advantage compared to standard fMRI is you have this map of activation that only need one trial and is showing an effect size change of like 0.75 because it's like 50% change. So you're looking at the blood flow, the oxygen blood flow, and you'll have a 50% increase for one trial of the signal, which is very strong compared to fMRI. And so they use this information to try to relate the pupil dilatation with the activation in the supplementary eye field and other regions. So the pupil dilatation is definitely related to the cognitive effort and novelty. And you can see here in these two nice scatter plot that as you're going through the experiment, the pupil dilatation is reducing in the case of the monkeys just because they're getting bored. But what essentially, you know, is being able to show, and here you have like the fuse imaging of the supplementary eye field. I'm going to try to magnify this like a lot blurry. Can you see it? Great. So you can see here this is a supplementary eye field, and those are like the score of a percentage of signal change. You see how strong the signal is here, okay? And here you can see the column and stuff. It's like pretty cool. I'm just gonna remove the, oh God, this is not well done. Trying to show you what's over here, but I got this, all right, I got the caption, okay. So if I do this, the caption's gonna disappear. Or should be disappearing. Yeah, great. So at the bottom right, you can see here they did a Pearson correlation between the pupil dilatation and the level of change in functional activation through ultrasound imaging in the supplementary eye field. And they use control regions and the anterior cingulate to check whether this was specific. And what they can see is like 30% of the variance of the pupil dilatation, so an external marker, is actually predicted by the level of activation of the supplementary eye field. So next, that's interesting because it's the first time we really show like such a strong relationship between something that is happening in the brain and really something that you can measure peripherally, such as pupil dilatation. And so what they try to do after that is to model whether, you know, like the activation was a consequence of the pupil dilatation, or if it was arriving before the pupil dilatation. And so I don't think they have a figure for that. Or is that this one? Yeah. And so what we can see essentially here is that pupil dilatation arrives after the activation in the brain, and that's where they derive the notion of causality between activation in supplementary eye field and pupil dilatation. And that's pretty much a paper. So you have like a great technology advance through this functional ultrasound imaging that allow you to measure directly a region that we know is related to gaze, but as for the first time be related to something new, which is a pupil dilatation here with a very, very strong statistical relationship. Boom, end of the story.
Thanks, Michelle. That's quite exciting. Are there any questions?
Yes, just one. Do they still need to do a cranial window to do the imaging with the ultrasound?
So I spoke to Pierre, it's like it's our colleague in Paris. So they can do it with or without, and that depends on the resolution that you want to have. I think for this kind of like functional imaging, you got to do it with a cranial window. But in the future, with better models, I'm sure we can do it without. That's actually eventually what will replace MRI one day.
Hopefully.
We're gonna have those like helmets, you know, like in Star Trek.
That would be great, actually. Yeah. Not
You
to...
got a brain disconnection in the right hemisphere.
Oh no, it was just a misalignment.
Yeah. Any
So-
other question? Yeah.
No one. Wow, that was a fast Neuroccino then this week. 4 papers and we're bang on time. Fantastic.
Ooh.
Well done, everyone. Thanks for all the new people who presented this week. Very exciting. Keep the papers coming. Everyone else, you're welcome to just bring a paper and present it. You don't have to prepare a fine presentation like Ari did. It's fine to just bring the paper up and talk about it. I also wanted to let you know that you might want to check out our website, clinicalneoanatomyseminars.com, because we have a couple of talks lined up now for the rest of the year. So make sure you check the dates or you download our calendar so you have the dates ready in your diaries. And with that, that's all we have time for. Thank you very much for coming, for bringing the exciting papers, and we see you next Monday. Bye.
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 Bye.